Good evening and welcome to tonight's event. On behalf of the University of Southampton, I'd like to extend you all a very warm welcome to all of the staff, students, alumni and supporters who are joining us this evening. I'm Professor Mark Cragg. I work in the Centre for Cancer Immunology and one of the senior group leaders here. And tonight I'm delighted to be joined by two of my colleagues from the Centre, Dr. Tim Fenton, who will talk to us about some of his world-leading research and contributions to new research areas that he's been developing since we recruited him a few years ago. And also one of our postgraduate researchers, James Christian, who's going to talk to you about his current research project, what he hopes to achieve in that, and highlight what this project might mean for the future of immunology treatments. We're also joined this evening by Charlotte Moss, who's a clinical trial patient back in 2011. She's here to share her experience and to talk about her involvement with the Centre for Cancer Immunology. After their talks, we're going to have a Q&A session. And to submit your questions, please use the Q&A box, which you can see hopefully on the right of your screen. We'll hope to get us through as many questions as possible, so please feel free to enter them. And if we don't get to your question in time before the event closes, we'll still endeavour to respond to your questions. So without further ado, I'm now going to introduce Dr. Tim Fenton to present some of his research work. So Tim is an associate professor in cancer in Southampton. His research interests are focused on understanding cancer at the molecular level and how the disease tries to escape our immune system. As I mentioned, Tim was recruited to Southampton a few years ago, and he's going to introduce to you some of the innovative work that we're doing here in the Centre for Cancer Immunology. Over to you, Tim.
Thank you, Mark. Good evening, everyone. As Mark said, I'll talk to you a bit about some of the different work going on in the centre and how I think and why particularly I was attracted to come and work in Southampton and to bring my research here. So cancer, as I'm sure you'll all be aware, is a daunting but we think a surmountable research challenge. I'm going to talk to you a bit tonight about where we excel in Southampton, in particular using the immune system to fight cancer and how this has really represented a paradigm shift in the way we think about cancer and the way we think about treating cancer. And I'm going to talk about some of our own and some colleagues' work on understanding cancer as an ecosystem, what we call the tumour microenvironment, and how we can target various elements in the tumour microenvironment to kill cancer cells. And then I'm going to give a last slide on where some of the directions in which I think we're heading and where I'm particularly excited about progress over the coming years ahead. So the challenge, and this will come as no surprise to many of you, I'm sure, but it's a fact that 1 in 2 of us who were born in the UK since 1960 will go on to get cancer at some point in our lifetime. So clearly this is going to be a universal experience one way or another. But the good news is that since, if you think about the kind of modern era of cancer research, I guess, beginning in 1970, when the US government declared their war on cancer, at that point, 25% of us were surviving a cancer diagnosis for 10 years after that diagnosis. By 2010, thanks to cancer research, that proportion of people surviving long-term post-cancer diagnosis have risen to 1 in 2 of us. And Cancer Research UK, in their research strategy set out several years ago, are aiming to make that 3 in 4 of us surviving our cancer long-term within 20 years. So that's an ambitious target, but you can see how that progress really has been driven by cancer research and how we hope to improve that and eventually to almost all of us, or hopefully everyone, surviving their cancer long-term. So why was I attracted to Southampton? I moved my research here just over 2 years ago now, really because Southampton is a centre of excellence for cancer immunology. And cancer immunology has been a discipline for many, many years. And for a long time, it was an unfashionable discipline amongst cancer researchers and didn't receive as much attention or indeed as much research funding. But people, including many researchers in Southampton, worked a way to understand how our immune system responds to cancer for many years. And then just over really 10 to 15 years ago now, this really resulted in an explosion in our ability to treat cancer. This is a piece of data that continues to amaze me, really. This is a drug called ipilimumab, which is an example of a cancer immunotherapy. And what this drug does, it's an antibody, is it takes the brakes off our own immune response to cancer and allows our own immune system to kill cancer. And this just shows that the long-term survival of people with very advanced skin cancer, so metastatic melanoma. And it shows that although sadly, even with this drug, ipilimumab, at the time these trials were run, many of the patients would sadly still succumb to their cancer within the first 2 years. There was then a remarkable phenomenon occurring. Actually, the remaining 20% of patients, you can see this curve becomes a horizontal line around the 2-year mark post-treatment. And at this point, people are surviving long-term with their very advanced skin cancer that would otherwise have killed them very quickly. So ipilimumab was clearly doing something very spectacular. And this led to an explosion really in the press, a lot of press attention, and I'm sure many of you will have read about cancer immunotherapy. So really, as I say, coming out of almost a backwater of research for many years, immunotherapy came centre stage into cancer treatment, with its success initially in advanced skin cancer, in some lymphomas, and also more recently in other cancer types, including lung cancer. And this resulted in the Nobel Prize being given for cancer immunotherapy several years ago now. So cancer immunotherapy is really the exciting place to be in cancer research in 2024. And in Southampton, we are pushing ahead with advances in cancer immunotherapy in the 3 main domains of cancer immunotherapy. So we have antibody therapy, and Mark, who introduced me, is a leading world expert in developing antibodies to target cancer and to activate parts of our immune system to target cancer. We're also working on strategies for improved vaccination against cancer, so therapeutic vaccines. Since COVID, we're all very aware of RNA vaccination and then the potential of RNA vaccination. That's applicable, we think, in the cancer context. And there are a lot of efforts underway to vaccinate against cancer in patients who have been diagnosed. And then checkpoint inhibition. And the drug I showed you, ipilimumab, the antibody, is an example of an immune checkpoint inhibitor. One of these drugs that can take the brakes off of our immune response to cancer and allow our T cells to attack cancer cells. And the important thing to note about immunotherapies is that compared to conventional chemotherapies and radiotherapy, which can have extremely nasty side effects, as I'm sure you're aware, oftentimes the cancer immunotherapies have fewer side effects that are generally milder. They're certainly not without their side effects, but they're generally milder and easier to manage. So just bear with me. How do we do good science in Southampton? Well, one of the things that we have here is a really strong connectivity with other parts of the institution. Particularly, we're based at the hospital site in Southampton General Hospital, and the Centre for Cancer Immunology is part of the wider School of Cancer Sciences spread across 3 buildings. But we're also very connected in with the clinical trials unit in Southampton, also with the biology departments, engineering over on the main campus, and with the other elements in the medical school, which really allows us to exploit our basic research discoveries and bring them into the clinic. I'm going to tell you a little bit about a couple of stories in which this has been done in Southampton in recent years, but I'm sure when I was showing you that previous piece of data that I was very excited about with ipilimumab, it won't have escaped your notice that actually I was talking about very good results for 20% of patients. But what about the other 80% of patients? Clearly, we have a long way to go, because what we want to be doing is for that line to be a horizontal line at 100% of patients surviving their cancer long term, not 20% of patients. And at the moment, there's a lot of work to be done in understanding what makes that 20% of patients gain a good response to ipilimumab? Why do those 20% of patients get essentially cured of their skin cancer while the other 80% don't derive this benefit? So that's really where a lot of our efforts, and particularly this is work in a number of the labs in the Centre for Cancer Immunology, including my own, is trying to understand if we can look at a patient at the time of diagnosis or in their tumour material and understand whether that patient is likely to gain benefit from our immunotherapy, or which immunotherapy they'll gain benefit from. The last thing we want to be doing is giving expensive treatments to people who will gain no benefit, and actually they will still potentially get side effects. So we want to spare people treatments that won't help them and give people treatments that will help them. So I briefly mentioned T cells. T cells are really a key weapon of the immune system against cancer. T cells are white blood cells. So they're involved, particularly effective at responding to viral infection, for example, but they also can attack and kill cancer cells in using very similar mechanisms as the way that they attack and kill virally infected cells. Now, I've talked already about ipilimumab. Ipilimumab is an example of a checkpoint inhibitor or a checkpoint blocker. And there are a number of antibodies shown here on the right, like ipilimumab, that have been approved for treatment of various cancers in humans. And what ipilimumab and these related antibodies on the right-hand side here do is they actually flick off switches that are on the T cell. So T cells are very potent weapons. They'll attack virally infected cells, they'll attack cancer cells, but they can also, if they're not regulated properly, they can attack our own healthy cells. And this is the cause of autoimmunity. So the immune system is a very powerful weapon and it needs to be very carefully controlled. So normally these immune checkpoints, things like CTLA4 and PD1 that I've shown on the right here, are off switches on T cells. And in cancer, the cancer can actually engage these off switches and shut down the T cell response to the tumour. What these immune checkpoint blocking antibodies do is they relieve this inhibition. So they stop cancer from flicking the off switch and they allow the T cell to remain active against the tumour. Over on the left, we have a different set of switches on the T cell, and these are switches that help to potentiate the activity of the T cell. And here I'm going to talk to you about a little bit of work that's been, or a large piece of work, led by a clinician scientist in the Centre for Cancer Immunology, Dr. or now Professor Sian Lim, who's a lymphoma doctor and researcher in the department. And she did a large body of work, starting with mouse models and progressing all the way through to a clinical trial called the REVA trial for patients with B-cell lymphoma, where she added an antibody that actually switched on a switch called CD27 on T cells to activate T cell responses against lymphoma. And this was combined with an existing antibody used to treat lymphoma against a molecule on the B cells themselves or on the lymphoma cells called CD20. And a lot of work on understanding the biology of that target, CD20, has been done within the Centre for Cancer Immunology or in the department previously by Mark and others. So that's an example where the entire rationale for understanding why it would be useful to flick the CD27 switch on T cells in addition to targeting CD20 on the lymphoma cells was all worked out here in the department, in the Centre for Cancer Immunology, leading to this REVA trial for B-cell lymphoma. And now that's promoted yet another round of research in which Sian and colleagues in the CCI are developing even more effective CD27 antibodies, which act to engage that CD27 switch and turn T cells on to allow them to attack the lymphoma cells. One other piece of research I'd like to tell you about briefly this evening involves a collaboration is being led by an experimental pathologist in the department, Professor Gareth Thomas, And this is an example of a treatment for head and neck cancer. We've collaborated with Gareth and his team for several years, and these data on the left here are taken from a paper that we published with Gareth's group a couple of years before I came to Southampton. Now, this is a rather busy slide, but I'll talk you through the main elements of it. I mentioned earlier the cancer ecosystem, the tumour microenvironment. When a cancer is taken out of a patient, that's not just a ball of tumour cells, it's a ball of tumour cells, but intermixed with that are all kinds of other cell types from our own body that have been kind of co-opted into the tumour. So blood vessels, for example, immune cells, stromal cells. So there's a lot of different tissue in that tumour. And on the top left, those four pictures you can see at the top left one is that those brown dots there are actually T cells that are in this tumour. So that you can see in parts of the tumour, they get full of T cells. And that's why if we can take the brakes off of the T cell responses, sometimes we can get a very strong anti-tumour response. These are T cells that have gone in to attack the tumour, but the tumour is suppressing their activity. Other tumours, like the one immediately below the picture with all the brown dots, has very few T cells in it, but what it has lots of, and we found this with Gareth, is that it has lots of a protein called smooth muscle actin, that SMA, which is the brown stain there in the bottom right picture. And that is staining, essentially scarring of fibrous tissue that's formed in the tumour. So what Gareth's lab showed through a series of very elegant experiments in cell and mouse models is that if you have tumours that have a lot of this scarring in the tumour, that builds up a network of what we call collagen that keeps the T cells out. So if you have a lot of scar tissue in the tumour, you can't get the T cells into the tumour. So with this in mind, Gareth and his team devised an alternative way in which to treat tumours. And they showed, as I say, using animal models, that actually, if you can target the scarring process, so the cells that mediate this are called fibroblasts. If you can target the fibroblasts, so we're not trying to target the tumour cells directly now, we're targeting the fibroblasts in the tumour, you can actually melt that scarring away and allow the T cells into the tumour. Then when you use your immune checkpoint blocking antibody to activate the T cells, you get a very effective anti-tumour response. So you don't just need to switch the T cells on sometimes, you actually need to get the T cells where they need to be in the tumour and then switch them on. And so this is really exciting work. And this has led to a clinical trial in head and neck cancer. And this is a phase 2 clinical trial. So the safety of the drug being used is already known. So we've gone straight to trying to show how effective it is. So I just very quickly, because I'm running short of time, just take you through a couple of areas in which I think things are going to be progressing in the coming years. Getting a really deep understanding of how our antibodies work at the atomic level resolution, improving the prediction of the peptides that we use to design better vaccines. These are all things happening in the CCI. Generating better cell and animal models for understanding the interaction between cancer cells and our immune cells. Improved DNA sequencing to understand how tumour cells differ from our own immune cells so we can target them better. And understanding, as I say, the tumour ecosystem. So really building up that picture of all the different cell types in the tumour and how we can modulate their behaviour to get better therapy. And this is all relying on collaboration between ourselves, but also increasingly across the disciplines and with industry partners, and also trying to look into, of course, preventing cancers. And there's work going on in the CCI on that front as well, particularly with a major trial in prevention of lung cancer. So I thank you for your attention, to answer questions later and leave it there. Thank you.
Fantastic. Thanks very much, Tim. So hopefully Tim's given you a bit of insight into the type of work that we're doing in the centre, about how many people are involved in each study, and the fact that we try to bring together clinicians with basic scientists and scientists across multiple different sites, both at the university, nationally and internationally. So it's a complex area, and it involves a lot of people to try and make good progress. So obviously to continue to make progress, we need young people to come into the field. And our next speaker is going to be James Christian. So James just started his PhD at the centre in October. So he's in his first year of his PhD. And his PhD really gets at the heart of some of what Tim spoke about, which is really trying to understand why immunotherapy works well for some people, but not so well for other people. And just to recognise that actually James's PhD is funded by donations to the Centre for Cancer Immunology, the Cancer Immunology Fund. So without further ado, I'll hand over to James and he can explain what he's been up to.
Thank you, Mark, and good evening, everyone. As said, I'm James, and I started my PhD at the CCI just at the end of last year. But the truth is that I've been on this path for just a while longer. You see, when I was a kid, I wanted to be a mad scientist when I grew up, though very much the mad part was optional. Simply, the world around me and how it worked fascinated me, and I always wanted to know more. That took me through secondary school and ultimately to a degree in natural sciences at the University of Bath. But even at that point, I didn't really know what I wanted to focus on, not until I attended a talk, an immunology talk by the University of Southampton. There, I learned that the immune system can fight cancer. We've already heard that today, of course, but the key part is that this is normal. You see from this video here, there's T cells tracking down and killing cancer cells. And your bodies do this every day. Even whilst we've been talking today, your bodies have probably killed a cell that could have become cancer. The problem is when cancers learn how to hide. Immunotherapy as a field helps explore why this happens and aims to empower and engage the immune system. Of course, when I heard this, I wanted to be involved. And as soon as the talk ended, I asked for a tour and ultimately a year of work experience at the Centre for Cancer Immunology. There, in 2019 and 2020, I met many of the colleagues I work with today and learned an incredible amount about cancer and how the immune system works, though ultimately my time there was cut short by the pandemic. So I popped off back to Bath, and even then I was already planning my research and seeing how I could move things forward. Though for all I'd learned about cancer, that was all quite abstract, really. And pretty soon after, my parents were diagnosed with cancer. First, my mum, and then my dad. And that's something you don't really see in textbooks. The pain of diagnosis and the sort of burden of chemotherapy and the long-term side effects afterwards. So when I was offered funding to do my PhD, charity funding, I knew then that I wanted to work on new approaches to curing cancer, because I believed that immunotherapy could do better, produce new treatments that are not only more effective, but kinder as well. And immunotherapy has already come a long way. Tim's already talked to you about immune checkpoint proteins, which is one of the most common ways that cancers can hide. These two proteins, CTLA4 and PD1, are two of the main culprits, preventing the activation of new immune responses and deactivating existing ones, leading to this sort of suppressive zone around tumours where it's hard for the immune system to get things done. And immune checkpoint inhibitors, the drugs that Tim mentioned, these antibodies, can reverse this effect, instead promoting immune activation and sustaining immune responses. And we have seen miraculous recoveries with this kind of drugs. Here, a patient with a particularly massive tumour just by their neck. You can see from these pictures, the big lump, it shrinks in just a matter of weeks. And when it was removed by surgery, it didn't come back even 3 years later. The problem, as Tim mentioned, is that this doesn't happen for everyone. And one of the key factors is that there needs to be immune cells ready and waiting to be activated. Otherwise, it's like starting a race with no runners. And the immune system has its job cut out for it. I've got two pictures here, to sort of spot the difference, to sort of show what the immune system is looking for. It's looking for small differences in cells that might indicate that it's cancerous. This one is hopefully quite easy. I can't point it out to you, but it's a purple jelly bean just near the middle on the right. But unlike these images, where there's about 100 cells, the body has trillions, and the differences can be very small, which is why we need to train the immune system. And one of the key tools for doing such is a vaccine. Vaccines usually have a few components. The main one, the first one, is that we need to give the immune system a target. We call this an antigen. This is usually a protein that cancer cells produce, but that healthy cells don't. If we can target that and teach the immune system to recognize it, then it can track down cancer wherever it might be, even once it's spread. The second important feature is a trigger. The immune system needs a reason to fight anything. It needs to see it as a threat. You can consider this as like calling 999. Emergency services don't show up unless they're called. Finally, repeat exposure is important. The more we can teach the body that these two things, the threat and the target, are linked, the more it will learn how to fight it. And ultimately, you end up with immune memory. And immune memory cells are special because they are essentially fully trained, sort of James Bond license to kill. And this means that they can ignore a lot of the immune suppressive effects we see around tumors, making them very good at targeting cancer. This sounds lovely in principle, but there's a few caveats. Unfortunately, the best target is often different between different patients and different tumors. And trying to find out what these targets are is expensive and difficult. Secondly, if we want to build immune memory, that takes quite a long time. And we don't want to give time for cancer to evolve and develop. Which is why, with my research, I'm aiming to make a cancer vaccine, but in a way that you might call cheating. You see, there's already plenty of the best possible targets in the body, in the tumor itself. And secondly, any immune response near to a tumor can help trigger an anti-cancer response if it's strong enough. This is because any immune response in progress releases danger signals that can interfere with the cancer's ability to hide. This sort of makes sense in theory, like where it'd be easier to hide, an abandoned warehouse or that same warehouse as it's being searched by the police. And indeed, you often see in the news sometimes, like someone's infection causing a tumor to shrink. And it's one of the ways that we try and treat, in particular, bladder cancer already. So we know that immune memory cells are good at attacking tumors. And we know that an immune response in progress can help take away the invisibility cloak of cancer. So what immune memory response could we use? Well, we've already got plenty of memory cells in the body, and some were produced very recently. So I think I might have given away from the title slide, but over 94% of people in the UK have received at least one vaccine against COVID-19. And people most vulnerable to cancer, ages 60 and up, often receive many. So I think that this is something that we can use, if only we can direct it at the tumor itself. In my research, I plan to take SARS-CoV-2, which is the virus that causes COVID-19, and take out the harmful components, essentially making a cardboard cutout that the immune system can recognize. And I plan to combine this with other immune activating drugs and inject it directly into the tumor. The aim here is that I will teach the immune system how to fight cancer by having it train on the best material available, the cancer itself. And in that way, because you're training it on the cancer, it doesn't matter where it is. Whereas a lot of vaccines tend to focus on one particular type, this, in theory, might be able to help treat many. As a sort of recap, I've got another spot the difference exercise, but this one's a little bit harder. You see, whilst cancer might hide in the dark corners of the body, such that the immune system can't see it, from the outside, we know exactly where it is. We can see it on scans. So instead of training up the immune system and letting it find cancer on its own, I instead plan to tell the body exactly where the problem is, in a way that it can't ignore, like an explosion in a haystack. So if I take away this image now, can you spot the difference? Thank you for your attention. Thank you for listening. And I hope to hear some questions from you later.
Fantastic. Thank you very much, James, for a really clear presentation and description about what your project hopes to do. And so hopefully, I think James will have conveyed to you the idea of continually evolving new ideas about how we can use the immune system to treat cancer. So in this case, using SARS-CoV-2 as a potential vehicle to be able to do that, and the idea of vaccinating against cancer. So all of those are really intriguing and interesting ideas that we're going to try and explore over the next few years. So thank you very much, James. And so we'll move on to our final speaker for tonight, which is Charlotte Moss. So Charlotte was diagnosed with melanoma back in 2011, and she was told her cancer was not treatable by chemotherapy and radiotherapy. She then decided to take part in a clinical trial on a new experimental cancer treatment called immunotherapy. And as Tim mentioned, perhaps 10, 15 years ago, it wasn't so commonly spoken about as it is now. And so 10 years later and cancer-free, Charlotte's here to tell us a little bit about her experience with cancer and her involvement with the Centre for Cancer Immunology. Thank you very much, Charlotte.
Thank you very much. And thank you for inviting me to come along today. Before I tell you a little bit about myself, I wanted to ask everyone, essentially, what is your lucky number? Apparently, the most popular response to this question is 21. I was a bit surprised by that. Or maybe you might be selecting the lucky number 13 as your lucky number. If somebody asked me that question, I would say, rather unusually, 1,200. And the reason for this is that was my patient number on the clinical trial at Southampton. It was also the total number of people who were on that trial. So I was the very last person to be accepted. And so for me, it's become a very exceptionally lucky number. When I was first diagnosed with melanoma back in January 2011, I had a very small nondescript mole on my knee that I wasn't seriously concerned about, but it had changed slightly, so I got it checked out. Had it removed, and unfortunately was confirmed very quickly as being melanoma. When I was told this, I took a deep breath, but because the mole was so small and it didn't seem like massively significant, there was no huge amount of pain or I didn't feel ill, my initial response was actually quite a little bit kind of positive. Like, what do we do now? What next? There must be something else. And actually, it turns out what next was 2 more surgeries. I had a wide local incision, which was to take a larger area of the original site, and a sentinel node biopsy, which is essentially part of the immune system. The surgeon described it at the time as almost like a knight with a little kind of a sword or a spear, just kind of looking after the body or in part of your immune system. But unfortunately, when they took out the sentinel node and they undid the main layer, there was no longer any node, there was only cancer. And it was really at that point that it hit me, that it wasn't just a mole that had started to change and look a bit different, but the cancer had spread. And the results really showed that it had been spreading for quite some time. Another surgery followed. This was called a block dissection, where quite a few sentinel nodes, quite a few nodes at the top of my leg were removed. Thankfully, at that point, no more cancer cells were found. But that did mean that I was very advanced stage 3 cancer. And the odds at this point in time were quite poor, just 20% chance of surviving the next 2 years. I thought at that stage that there would be the more traditional treatments of radiotherapy and chemotherapy. But I was told that given the seriousness of where I was, that these would not be effective. And the only thing at that point in time was to wait and see if the cancer returned. And it's a really odd feeling knowing that there may be cancer cells inside growing, but there's nothing you can do and you just have to wait until it starts to become obvious. However, here is where my lucky number finally came up. I was invited to join a clinical trial that was being run out of Southampton, which at the time wasn't anything I'd heard of before. It was a new type of immunotherapy drug, a new type of treatment. And one of the main reasons I wanted to join the trial was because it was a very clever solution. I just thought it was really great to be able to use your own immune system, give it a bit of an upgrade to recognize the melanoma and deal with it. It just seemed almost a bit obvious in some ways, but it just seemed such a unique way of dealing with it. So I started a 3-year clinical trial. The treatment itself was really straightforward, especially in comparison to the surgeries. The bulk of the treatment would be a trip to hospital every 3 months. On a Friday, I'd have an infusion for 90 minutes. Actually, the worst part was trying to get the cannula in, especially in the winter. I would then take it easy over the weekend, and then I was back at work on Monday morning. And it really was as simple as that. And it really helped me take back control. There was no visible external signs I was going through any treatment. At work, I didn't have to explain what was happening. I didn't have to take large periods of time off work. It was all very normal. Every 3 months, I just happened to take a long weekend, and that was it. So since the trial, I have now been cancer-free for over 10 years, which is incredible, actually. And I'm now really looking forward to continue to work on my hobbies and my drawing. And when I was first diagnosed, I was very focused on the very short-term view. So just trying to get through that day, or just trying to get that week or that month. And now I'm thinking about what I'm going to do for the next 5 to 10 years. What are we going to do when we retire? Kind of the normal conversations that you would have. And since the success of the treatment, I really wanted to give something back. And so speaking to the team at Southampton, they suggested I get involved as a patient representative in the clinical trials unit. There is a group of us who meet on a regular basis, and we are presented with a number of clinical trials, and we just comment and see what it's like as a patient, and we try to kind of put that voice of the customer, voice of the patient across for the clinical trial. And it really does feel like you're making a positive impact. It's really good. It feels that my experience can be useful and be able to give something back and help others in their journey. It's also quite nice to think that someone would have reviewed the clinical trial that I was on. I'm not sure if it had the same structure in place, but someone would have been checking if the forms were clear, that it was easily understood, that the patient on the trial would understand the process, and so on and so forth. And when I signed up for that, somebody would have done it. So it feels quite nice that it's kind of feels like it's going full circle. So being part of the group really does bring home the huge number of people involved. Obviously, we've heard today how many people are involved in kind of doing the research, but there's a huge number of people that sit behind the scenes who get involved in developing and creating new treatments. And although I've kind of mentioned about being lucky, that really had absolutely nothing to do with it. It was the hard work and dedication from everyone here today, and everybody back at Southampton. They're the people that saved my life. The only lucky part was that I was connected to Southampton, and then it is the centre of excellence that it is, and long may that continue. Thank you.
Thank you very much, Charlotte. That's a fantastic to hear about your experiences, and thank you very much for your endorsement of our centre. So that concludes our speakers for the evening. And so what I wanted to do is just take a couple of minutes really to hopefully summarise a little bit about what you've heard. So the centre opened just over 5 years ago in April. 5 years ago, and since then I think we've come a long way. We have over 100 people, scientists, basically working in the building, and just to recognise that the building itself was funded entirely by philanthropy. So the building wouldn't be here without the generous gifts of donors. And really, what we've tried to do in those 5 years is, build an infrastructure, some of the things you've heard about from Tim and James, where people can come and work on cancer immunology in an environment that allows them to really work at the cutting edge. We provide some of the best facilities. We have very good scientists, we have lots of good infrastructure around us, which allows us to really ask the difficult questions in cancer immunology. None of that happens alone. As we mentioned, we're just across the road from the hospital. And so we work very closely with the NHS, our clinical colleagues. And one of the key things I think over the last 5 years has really been the recognition that actually working on primary human samples, which patients donate, that are removed from surgeries and people give permission for us to work on. And that's absolutely crucial for us to understand really what cancer is and how we can go about making better treatments for it. And so thank you to the patients that donate their samples and also to the interaction that we have with the NHS. And then Charlotte mentioned it, the clinical trials unit here in the centre. It's a fantastic clinical trials unit. They're developing clinical trials, both the kind that Tim mentioned, which is what we call a phase 1 trial, where we take something that we've observed in the lab and try and test it for the first time in patients, in humans. And then also what we call phase 3 trials, which is where we're trying to see if our new immunotherapies are as good or better than the current gold standard for treatment. And sadly, in some cases, to be better than the current best treatment is not very good. And Charlotte alluded to it, the survival rate at the time she went onto her trial was 20% of people in 2 years. So, and Tim mentioned it as well, which is we want to move that curve basically higher and higher with an increasing knowledge of what cancer immunology and cancer immunotherapy treatments can do. And so really that's what the centre is. And thank you to those of you that have donated and been interested in the centre to date. And I think now we'll move to our Q&A session. I think there's been a series of questions that have been put in the chat. So I will take them essentially from the top down. And I will ask James to take all the difficult questions, Tim to take the second most difficult questions, and I will try and take the easy ones. Charlotte, feel free to take any ones that you'd like to. Okay, so the first question is, do some cancers first successfully treated by immunotherapy evolve to evade the immunotherapy treatment to then potentially reoccur in this evolved form? So that's a really great question. And sadly, the answer is yes. What we see when we use lots of different types of immunotherapy is that we might see an initial good response that the tumor might shrink. But in some patients that then after a period of time of receiving the immunotherapy, the tumor returns. And I'm sure all of you have heard of Charles Darwin and the theory of evolution. Essentially what that says, if you apply a selective pressure to an organism, it will basically, there'll be an evolution that it tries to basically survive that selective pressure. And the same is true of cancers on an individual single cell level. Every individual cancer cell is trying to survive. And so in some cases, we have effectively what becomes a race between the immune system and the cancer cell. The immune system is capable of recognizing some of that evolution that the cancer can do, but at the same time, the cancer is able to suppress the immune system. So it's really whether our intervention can sufficiently augment the immune system to allow it to eradicate the tumor before it can evolve away new defense mechanisms of the immunotherapy is really what the kind of race that we're in that kind of context. Tim, James, I don't know if you wanted to add anything else to that one.
Just that it's one of our major research interests in my lab is how that process of evolution happens. And we study some proteins that exist in cancer cells that are normally there to mutate viruses to stop them infecting us and to stop them being able to replicate, and they can actually mutate our own DNA in cancer. And our hope is that by understanding these enzymes, we may be able to block their activity and therefore limit the cancer cell's potential to evolve through gaining more mutations during treatment. So it's definitely a really important area and a big interest to us, certainly. Yeah.
James, did you want to add anything?
Just a small detail about how the immune system can target different targets within cancer, as I talked about. and some of those are much harder to mutate than others. So if we can discover which ones are common to cancers and cancers tend to keep them over time, then that can help us develop more robust immune responses.
Great, thank you. So if we move on to the second question, and that is, what impact does immunotherapy have on those with autoimmune diseases? So again, really great question. And I think at this point, it's probably useful to kind of split apart the idea that immunotherapy is one thing. Conceptually, it is one thing. It's using the immune system to fight cancer in this context. But actually, there's lots of different ways that we can do that, and they work in slightly different ways. So the antibody that Tim spoke about, the CD20 antibody, works by deleting the tumor cell. So we have a cell in the body called a macrophage, which is able to actually physically engulf and destroy tumor cells. Whereas the CD27 antibody Tim mentioned is what we call an immune modulator or an immune stimulator, which stimulates T cells and another type of cell called a natural killer cell. And so you can probably imagine that those two antibodies might work in different ways. And because of that, they stimulate the body and the immune system in different ways. And so they might have different impacts on the body in general. And so immunotherapy, in the context of people that might have autoimmunity, it's very much a case of what immunotherapy and what type of immunotherapy. So for instance, actually, the rituximab antibody that targets CD20, that targets B cells, is actually used for the treatment of certain types of autoimmunity. So it's used in rheumatoid arthritis and certain other autoimmune conditions. So in that context, it's very beneficial for someone with autoimmunity. It's actually used as the treatment, whereas you might not necessarily go ahead and try to stimulate the immune system with a T cell agonist or an immune modulator like CD27 that Tim mentioned. Similarly, with the checkpoint blocking antibodies, again, whether you would necessarily go in that context where there might be an ongoing immune response, such as in autoimmunity, yeah, you probably wouldn't use those in that context. But it really comes down to the type of autoimmunity, whether it's B cell mediated, T cell mediated, or otherwise, and the type of immunotherapy that we're talking about. Tim, did you want to add anything to that one?
No, I think we covered it very well, Mark.
James or Charlotte, any additional context for those ones? Okay,
Not
great.
for me.
No, that's great. The next question is, how do we join immunotherapy trials? So really good question. I mean, maybe Charlotte, if I throw that one to you first, how did you join your immunotherapy trial?
Yeah, I was invited to apply. So there was this clinical trial running, and I kind of fitted the overall cohort of people that they were looking to recruit for the clinical trial, and it was specifically for people with stage 3 melanoma to try ipilimumab. But it was brought up to me, it was mentioned by my surgeon at the time and the oncologist who kind of were treating me with more traditional treatments at the time at Southampton. And so I spoke to them about it and got their view and opinion, and took away quite a large amount of the material actually to understand what the impact would be, what it would mean to me, and how it would impact my life, and whether I really wanted to go on that journey. So that's how I got involved and got invited to join the trial.
Yeah, thank you. And I think that's pretty common. I think normally it's at the recommendation of your clinician. That should be your first port of call, really. They'll know from you the specifics of your particular tumor and the stage and everything else, whether you're suitable for a trial that might be running nationally or even locally. So I think that should always be your first port of call. Okay, next question. Why does immunotherapy only work on some types of cancers? So that's a great question. And one of the things that I think all of us that are working in cancer immunology are interested by. One of the determinants Tim mentioned in his talk is whether you have T cell infiltration, whether you have lots of T cells in there is one of the things that could determine whether you might respond to a certain type of immunotherapy. Another thing is how many mutations you have, which again relates to things that both Tim and James kind of are working on. So mutations are basically the thing that makes the difference between our host cells, our normal cells, healthy cells, and the tumor. And each individual tumor from an individual will be different from another. And actually within an individual's tumor, the genetic mutations will be different between each tumor cell in that individual's tumor. There's a huge amount of complexity, but certainly the correlation with immunotherapies has been the more mutations you have in your tumors, the easier it is for the immune system to detect and potentially destroy it. There's a good correlation with the checkpoint blockers, anti-PD-1 and ipilimumab and antibodies like that. That there's a good correlation there between genetic mutation and response, also with whether or not you have T cells infiltrating your tumor or not. So they're just a couple of the things. In terms of the tumors themselves, although each individual tumor is different, certain types of tumors tend to be more or less infiltrated with T cells. So that's one of the things that can determine it. There are also certain tumors that have particular mechanisms of immune suppression that are very common. One of the things that Tim mentioned that Gareth Thomas is very interested in is a cell called a fibroblast. That's a cell that actually can wall off a tumor and stop the T cells getting in. So if the particular tumor type has lots of fibroblasts, you can see how that maybe then the T cell therapy might not work so well. Tim, do you want to take over from that one?
Sure. And I think that the things that Mark has mentioned are state of knowledge at the moment, and it's an area in which we really have a long way to go, I think, understanding which tumor. So although there are some general rules of thumb emerging, if you like, so generally a tumor with more mutations will respond better to immunotherapy, and tumors that are full of T cells are likely to respond better. It's not the case that an oncologist could look at someone's tumor and say, you're definitely going to gain benefit from the therapy I'm about to give you on that basis. We just don't understand enough of how that all works yet. So that's a really intensive area of investigation at the moment, and will continue to be so over the coming years, to try and work out which tumor is going to. Obviously, you don't want to while you want to spare someone a therapy that's not going to work for them, on the flip side, you certainly don't want to withhold a potentially useful therapy from someone just because they have maybe a low tumor mutation burden or not much PD-L1 expression on their tumor. So getting these biomarkers right is really, really important to understand. So that's a major focus of research.
Yeah, thanks, Tim. So maybe we'll move on to the next question, which is, why do some people not tolerate immunotherapy? And I think that really kind of relates to the point I was trying to make about the autoimmunity. Every individual is different. So the way that your body responds to immunotherapy, to immune stimulation, is different. Sadly, we've all seen that through COVID. Different individuals respond differently to the vaccination. People were given the same vaccine, and yet their responses to it were very different. So both in a good way, in terms of some people generated very good COVID responses, but also some people had detrimental responses or didn't respond very well. That's all down to the individual genetics of every person. So there's a number of things that can play a role in whether or not someone can tolerate or not tolerate a particular type of immunotherapy. And again, it might depend on the type of immunotherapy. You might be able to tolerate a rituximab type antibody, but not so much an ipilimumab, or you might be able to receive CAR T cells, which we haven't really spoken about much this evening, but that's another type of immunotherapy. But again, another patient might not be able to tolerate that. Generally, the fitter and healthier you are, then the better you're able to tolerate cancer treatments in general. and to a certain extent, that's true for immunotherapies. Did anyone else have anything to add to that one? Great, so we'll move on to the next question, which is, do you think immunotherapy treatments can be developed for all cancers, or does it depend on where in the body it is? So I'm by nature an optimist, so my answer to that is yes. And just simply from a point that Tim made, which is really, we're at the start of our understanding about how cancer and the immune system kind of co-evolves and how it coexists. And James mentioned that when you actually see a tumor develop, it's already evaded the immune system, right? So all the time your immune system is finding and deleting tumor cells or potential tumor cells. And so I think the idea that if we understood that process better, and if we understood how the tumor cells and their tumor microenvironments suppress the immune system, then we can make therapies that allow us to overcome that. And checkpoint blockers and other therapies are our current best ways of doing that. But it's clear from increasingly sophisticated studies that we're doing now that this evolution of the tumor under selection from the immune system is finding ways of trying to evade it. And so it's clear that we need to understand things in a much more complicated way than we used to. At the start of my career, we used to look at a single receptor and a single signaling pathway. We're now in what we call an omics era, where we're trying to understand the signaling pathways that are happening in the cell all at the same time. So we have 20,000 genes, and each of those can be on or off. It could be at a low, medium, or high level. So if you think about that as a system that we need to understand, it's incredibly complicated. But I think with enough effort and time, then I think we can understand it. It's not ultimately, it's not a magic box. It's a system that responds to stimulus and suppression. So I think we can find ways eventually of being able to treat these different types of cancers. The question about where in the body it is, and I see there's a question a little bit lower, which is about crossing the blood-brain barrier for brain cancers. So that's a really interesting question, and that's so generally immune cells don't cross the blood-brain barrier, the T cells, very, very frequently. But actually people are designing antibodies and other therapies that actually can cross the blood-brain barrier. We use a receptor that's on one side of the blood-brain barrier and use that as a shuttle to take antibodies across the blood-brain barrier. So people are designing those things. So really clever scientists in industry and in other academic institutions are using what they call things like the brain shuttle, which allows things to cross that blood-brain barrier. So I think we will be able to treat cancers that are in more what we call immune privileged sites. I see I'm getting a prompt to say this is the last question. What is the main challenge in immunotherapy research? So that's unfortunate. So that's going to take me at least 45 minutes to answer that one. But I would say the complexity. That would be my response. It's incredibly complex. It's incredibly dynamic. It responds and it changes. That would be my response. Tim?
Yeah, there's so much to understand still, and so many targets that we haven't even discovered yet. So yeah, there's a lot to be done. Yeah.
James, as a newbie at the start of your career?
Definitely the complexity again. Cancer, it's not one disease, it's hundreds, thousands, all with different best ways of treating it. And trying to find different ways of tackling this, a sort of toolbox that we could use in clinics, is quite a big challenge to tackle.
Great. Charlotte, did you have anything to add?
No, I didn't have anything to add, other than I'm just quite in awe of the work that you guys do. I think it's very impressive, and it's been I'm probably going to say something, but it's just been really informative, and to get an update on where the immunotherapy treatments have been going. Certainly since I started on ipilimumab, that was the only one, and it sounds like there's many available now.
Great. Thank you very much. So thank you everyone for joining us tonight. So that's the end of our Q&A. As I mentioned, we'll try and respond to the questions that are left in the chat. So you've heard from some amazing people, hopefully you agree, and that we're all coming together to hopefully try to find ways of fighting cancer in a better way. So obviously James and Tim are on the front line of that, developing new ways to diagnose and treat cancer. And obviously we wouldn't be able to do that without people like Charlotte taking part in clinical trials, because that's ultimately how we test what we're trying to develop in the lab. And obviously that happens with a fantastic clinical trials unit and partnership with the NHS, as I've mentioned. So the other person that's obviously important in cancer research is you guys. As I mentioned, nothing would have happened in this centre. We wouldn't have the centre without philanthropy. So that's through generous gifts of you. And hopefully some of you will consider making a donation to the centre tonight. You can make a donation by post, by phone, or by visiting www.southampton.ac.uk/donate. The details should be on the screen, and we'll circulate them by email after the event has closed. And in a minute, you'll see a short poll, which will come up on your screen, which we'd like to ask you to complete. So that's really just, we'll take a couple of minutes of your time and gives us some feedback on whether what you've heard tonight is useful, informative, if there are ideas you have about other things you'd like to hear about. And for any of you that would like to watch what we presented tonight again, we'll be sharing the footage over the coming days. So keep an eye on your inbox for that. And you'll also find details of how to join our mailing list so we can keep you updated on future lectures. So I've gone 1 minute over. I'm very sorry, but thank you very much for joining us, and good night.